Hello, Year 9. Welcome back to the plant unit. Last lesson we were doing some work on how does what are the limiting factors that affect the rate of photosynthesis. Today we're going to look at those in a little bit more detail and hopefully be able to answer the question, how do the factors affecting the rate of photosynthesis interact with each other? So again, this is your driving question. Please make sure that goes on the top of your page, get it underlined, put the date as well. This allows us to keep our work organized. And our keywords for today's lesson are photosynthesis, optimum, limiting factor, carbon dioxide, temperature, light intensity, concentration, just like last lesson. But also, we're going to be talking about the inverse square law today. Bit of maths, not easy stuff. Enzymes and denature. Okay, so some of you will know what enzymes are. Those are biological catalysts, proteins that living things use that help speed up chemical reactions. There are enzymes all over. There are enzymes in your cells. They help chemical reactions happen. Denature is what happens to enzymes if they get too hot. Basically means the enzyme stops being able to work. Okay, effectively kills it, except it wasn't living necessarily in the first place. And the inverse square law is a law, which is like a rule, looking at the relationship between two things. And inverse meaning as one increases, the other one decreases, and square is to do with, timesing it by itself. So inverse square law, enzymes, and denature are the new keywords for today. Okay, what are we doing? We're looking at the plants unit. This will help us protect our environment and improve the yield of crops by understanding plants better. Okay, as we totally rely on those. Your starter is using the clues: the fridge, the moon, well, the night time, and the this whatever that is. What would limit the growth of a plant? Why can't this plant grow? What do you think? Pause the video. When you come back, we'll go through some answers. Well, if a plant is too cold, if it is too dark, or there is too little carbon dioxide, then a plant can't grow. Fridge, too cold. Nighttime, too dark. Not enough CO2. This molecule, 1 carbon, 2 oxygens, that's CO2. Not enough of that would mean the rate of photosynthesis is limited, as temperature, light intensity, and concentration of carbon dioxide are all limiting factors. And this is what we looked at last lesson. Today we're going to look at how they interact with each other. Obviously, this is all in the context of photosynthesis. The reason that a plant can't grow is because it doesn't have enough glucose, which can be used for growth, also used for respiration. The chemical equation for photosynthesis is water plus carbon dioxide makes glucose and oxygen. The symbol equation for that would be 6 waters plus 6 CO2s makes 1 C6H12O6 plus 6 O2s. Okay, hopefully we've seen this quite a lot of times. And through repetition, we are getting it stuck into our long-term memory. Again, I've written light because light is one of the limiting factors. It requires light to happen. It is an endothermic reaction. Right. Last lesson, we found out that light intensity, temperature, and concentration of carbon dioxide are limiting factors on the rate of photosynthesis. How bright the light is getting to the chloroplasts will affect how quickly photosynthesis happens. The amount of CO2 the plant has access to and the temperature within the plant will affect the rate of photosynthesis. Today we're going to be looking at what would happen if we plotted that data. And there are 3 graphs, one for each limiting factor, that you are going to need to memorize the shape of. Okay, and then we're going to actually look at some data and talk about that later on. If we plotted graphs of rate of photosynthesis against each factor, what would they look like? So along the x-axis, you've got the limiting factor. So start at the bottom, that would be 0. Work your way up, that limiting factor gets higher and higher and higher. So for instance, down at the bottom, it could be no light intensity, complete blackness. Work your way up, getting brighter and brighter and brighter and brighter. Okay. Up the side is a measure of rate of photosynthesis. So at the bottom, no photosynthesis is happening at all. At the very top, the most amount of photosynthesis is happening. Okay. It's Doesn't have units at the moment, but that could be number of bubbles per second. As in the required practical and previous lessons, we could use that as a measure. Okay. So the graph for how light intensity affects rate of photosynthesis looks like this. Okay. How could we describe this? Well, to start with, there's a steep gradient. So what we're looking at here is as the light intensity increases, so too does the rate of photosynthesis. Does that make sense? So for the first bit of this graph, increase light intensity, you are increasing the rate of photosynthesis. But after a while, you notice that it levels out, and increased light intensity, so it still might be getting brighter and brighter and brighter and brighter, but this does not affect the rate of photosynthesis. It does not go up or down on the y-axis, even though we are getting brighter and brighter. You see, this is the relationship between light intensity and rate of photosynthesis. Steep line, then it levels off. Concentration of carbon dioxide is the same. If you increase the concentration of CO2, concentration could be a percentage of CO2 in the air that the plant is in. Increase CO2 concentration, you too increase the rate of photosynthesis. So to start with, more CO2 means more photosynthesis until you get to a certain amount of CO2, and then adding more does not affect the rate of photosynthesis. So some of you will be thinking, what is going on here? But actually, more CO2, more photosynthesis, levels off. The third limiting factor is a different shaped graph. So the first two are the same. The third one is different, temperature. So what we're looking at here. So along the bottom, the x-axis, increase of temperature as you go along like that. So to start with, there is a slow increase. As you increase temperature, you also increase the rate of photosynthesis. This is what we'd expect. We know that the higher the temperature of the particles, the more collisions they're going to have, the higher the energy they're going to have, and this will increase the rate of the chemical reaction. So that makes sense to start with. But after a while, after it gets to a certain point, more temperature actually decreases the rate of photosynthesis, which is why you get this up and down shape on this graph. And in fact, on the way down, it happens a lot quicker. So some of you will be thinking, why is that happening? And because it goes up to a point, you are able to draw a line down and see that there is a optimum temperature. Okay, so at this temperature here, which is for most plants at roughly 40 degrees C, although it does depend on the plant, obviously, there is a temperature at which photosynthesis happens the fastest. And if it goes lower than that, then rate of photosynthesis is going to be lower. And if the temperature is higher than that, it is going to be lower as well. In fact, even slightly higher, you're going to have a quick drop off on rate of photosynthesis. Okay, so those are the three graphs. Quiz time. Can I please ask you to match the limiting factor to the graph? You could do this on a scrap piece of paper. You don't need to write them down at all. I just want you to think which graph matches which limiting factor. Pause the video. When you come back, we'll go through an answer. Okay, welcome back. Here we are. So as two of them are the same, I've just put them together. Carbon dioxide concentration and light intensity have graphs that look like this, where to start with, more limiting factor means you've got high rate of photosynthesis until it levels out, whereas temperature, increase rate of photosynthesis until a certain point, in which case increased temperature then decreases the rate of photosynthesis. So the first two, it goes up and then along, and then temperature, it goes then up and then down quickly. Why do these graphs look this way? Difficult question. This, I suppose, is the explanation. You need to understand what the limiting factor means in detail, and then think, why will the amount of that affect photosynthesis? And in the case of temperature, why would too much actually reduce rate of photosynthesis? Well, these are the explanations. Rate of photosynthesis increases with increased light intensity at first. Then, increased light intensity no longer makes a difference. So that's a description of what we're seeing. This is because at this point, either temperature or CO2 concentration are the limiting factors. Again, I made a mistake here. That CO2, the 2 should be subscript. I wonder if that changed when I moved it across. CO2, that should be a lowercase 2. Apologies there. This is because at this point here, it's going to be another limiting factor that will be affecting it. It's obviously not too dark. That's not going to be affecting rate of photosynthesis. It'll be something else. It'll be one of the other factors. Maybe it's too cold. Maybe there's not enough CO2 available. Either way, it's not light intensity affecting it anymore, but rather something else. That's why it levels off. It's a similar story for carbon dioxide concentration as well. So again, rate of photosynthesis increases with increased CO2 concentration at first, then increased CO2 concentration no longer makes a difference. This is because at this point, either temperature or light intensity are the factors that are limiting it. So very similar stories between light intensity and carbon dioxide. Temperature is a little bit different, obviously, because it's got a different shaped graph. So to start with, increased temperature increases the rate of reaction all the way up to the optimum temperature. For most plants, this is around 40 degrees C, but it does depend on the plant. Please remember that. After this optimum, the plant can become damaged by the increased temperature. So imagine that, if you were to cook a plant in the oven, then obviously it's not going to be able to photosynthesize anymore because you've damaged the plant. Simple as that. If you increase the temperature, you're speeding up the chemical reactions until the point where you're actually killing the plant. And actually that's why it's so steep, because very quickly you're breaking the plant's ability to do this chemical reaction. The reason you're breaking its ability to do this reaction is because you are denaturing enzymes as well as damaging tissues. Now, enzyme and denature are two of the key words, two that we haven't really come across very much. That's the reason. But when you learn about enzymes and denaturing in a different unit, hopefully that will become more clear. Effectively, an enzyme is a protein that living things have that at high temperatures, the shape of that protein will change, and therefore its ability to help photosynthesis will stop. Okay, so those are the explanations. Please can you have a read of all of these boxes and then decide which one fits the answer to the question, which describes the graph for temperature. Should be a question mark there. Okay, please pause the video and decide which box is closest to the answer. Right, the answer is B. At first, high temperature means faster rate of reaction, but after the optimum, increased temperature reduces the rate of reaction. Okay, so the first one, although it is true, but only up to that 40 degrees, the optimum rate of reaction is 50 degrees C. That's usually too high for most plants. It's 40. And an increase in temperature means faster rate of reaction, then it levels off. No, leveling off fits more for light intensity or carbon dioxide reaction. It does not level off with temperature, but rather goes back down again. Okay. Now I'd like to introduce you to something called the inverse square law. So this is a rule that you can sometimes apply to things in physics or chemistry or biology, which will help you to be able to explain a relationship between two things. Now, this is something that you will see in maths as well as science, and the fact that we're linking maths and science together means that I'm going to rate this with a difficulty of around the yellow, the U section, you're using your understanding of science, using your explanation of science to explain what is going on between two things. Okay. The inverse square law can be used to explain the relationship between light intensity and distance from the light source. So we remember from our required practical, this lesson is directly linked to that one, when you move the lamp away from the plant, you are reducing the brightness. But the reason for that can be explained using the inverse square law. Okay, so it states that light intensity is proportional to the square of the distance of the light source from the plant. Okay, now obviously that's quite intimidating. Lots of you will be thinking, what on earth does that mean? But we're going to go through that now, and you'll have time again to get that into your heads. So it's proportional to the square of the distance of the light source from the plant. Okay, so forgetting the square for the time being, the inverse relationship, it's got an inverse proportion. What that means is, well, there is an inverse relationship between distance and light intensity. That's a fact. That's what we've seen. That's the idea that when you make the distance bigger, you make the brightness lower. Okay, so as the distance increases, the light intensity decreases. This is because as the distance away from the light source increases, the light energy becomes spread over a wider area. And by spreading over a wider and wider and wider area, you are going to reduce the amount of energy in that space. And that's what brightness is, I suppose. Okay, so as you increase distance, you decrease light intensity. That's what inverse proportion means. One thing goes higher, another thing goes lower. Okay, we've seen this diagram before. Light source starts here. R is going to be distance in this case, and 2R means twice the distance, and 3R means 3 times the distance. Okay, we measured the distance using a ruler, but we can see that the light intensity decreases because it's spread out over greater and greater areas. 1 square, at twice distance you've got 4 squares, and at 3 times the distance you've got 9 squares. This is the inverse square law. So, the inverse square law applies to the relationship between light intensity and the distance from the light source. It states that light intensity is proportional to the square of the distance of the light source from the plant. So what does that mean? Well, as you increase the distance, so that's 1d, one distance, 2d is twice the distance, 2d, and 3d is 3 times the distance, 3d. But notice what happens to the area. It's got an area at 1d, but as you double the distance, the area of which the light is spread over is actually 4 times. And because greater area means smaller energy per area, it's actually an inverse relationship. So it's 1 over, or you could think of it like a quarter of the energy is available at a point at 2D. At 3D, it's a ninth, or 1 over 9. Or you can see that the area is times by 9 if you triple the distance. The fact that it's an inverse square law means not only is that it's inversely proportional, so 1 over the number, but the number is actually squared. So double distance, 2 squared is 4. Triple distance, 3 squared is 9. Does that make sense? So the light intensity at twice the distance away is spread over 4 times the area. What will it be at 3 times the distance? Well, the light intensity at 3 times the distance is spread over 9 times the area. Does that make sense? So however many times the distance away it is, you do that number squared. And that's the area it's spread over. And so it'll be 1 over that in terms of its brightness. So the inverse square law applies to the relationship between light intensity and distance from the light source. It states that light intensity is proportional to the square of the distance of the light source from the plant. For instance, the light intensity at twice the distance is spread over 4 times the area, or 2 squared. The energy at 3 times the distance is spread over 9 times the area, or 3 squared. Now this is hilariously blurry, but this I suppose shows the relationship. So because it's inverse, it's 1 over, and the distance, or how many times the distance, squared. So twice the distance would be 2 squared, but because it's the light intensity is lower, it's 1 over 2 squared, or 1 over 4, so it's a quarter. 1 over 4. That's the maths. Let's have, so that's a lot of information there, I appreciate that. Let's look how it applies to some numbers. Okay, this is actually some data, and it shows the relationship a lot easier, I think, so this may help. What we've got here on this table is down the side, we've got the distance increasing, 10, 15, 20, 25, 30. Notice the distance from the light source goes up. Now we're looking at the rate of photosynthesis, which is directly linked to the brightness. And we can see that at a close distance of only 10 centimeters, we have 120. What happens as the distance increases? Well, the rate of photosynthesis, the number of bubbles per minute, decreases. So that's your inverse relationship. But in fact, what you might also notice is that the inverse square law applies. It doesn't just go down, but it goes down relative to the inverse square law. So if you were to double the distance, we should see the light intensity divide by 4. If you triple the distance, you should see the light intensity divide by 9. And then that will be the link to the rate of photosynthesis. So, for instance, between 10 and 20, you're doubling that, aren't you? Double distance, we should see a quarter rate of photosynthesis. And we do. 120 down to a quarter, 30. Triple the distance, say 10 up to 30, we should see that divided by 9. And again, we do. 120 divided by 9 is 13-ish. So, there we are. Between 10 and 20, which you're doubling the distance, divide by 4. 120 divided by 4 is 30. Triple the distance, go from 10 to 30, that's tripling. So what you do is you divide it by 3 squared, you get 120 divided by 9, that's 13.33, rounded down to 13. It works. Now, what I'd like you to have a go at doing is make predictions for the following distances. Some of you will be able to pause the video and have a go now. Otherwise, just hold on and I'm going to show you how to do this. You will have the opportunity to do this in our objectives right at the very end. Okay, so these are the answers. Notice that you are increasing, but not by 5 every time between 30 and 40 and 50. And 5 is actually lower than 10, which is why you don't see this, the relationship decreasing in the same way as before. How did we figure this one out? Well, 40, I worked this one out by doubling 20 up to 40. Okay, so double up to 40. So 20 centimeters up to 40 centimeters is doubled. So the rate must be divided by 4. So 30 divided by 4 is 7.5. That's how I did that. As it was doubled, the distance doubled, we did 2 squared, 1 over 2 squared, or 1 over 4 is timesing it by a quarter, which is a quarter of it, is 7.5. The 50, well, I just looked at doubling 25. So between 25 and 50 is doubled. So again, 17 divided by 4. Because doubling, the distance doubles. So 2 squared is 4, so it's a quarter, 1 over 4. So a quarter of 17 is about 4.25. The last one, bit more difficult. I actually worked backwards because we knew what 10 was. So this is the logic. 5 centimeters is half of 10 centimeters. So 120 bubbles per minute is 4 times smaller than the bubbles per minute at 5 centimeters. So if this one is going to be even smaller than the one at 5, it would be 4 times smaller, just like every time you double something. So double 5 to 10, you could make a mystery number 4 times smaller. But if we're working backwards, we could do this number 4 times bigger. And 120 times by 4 is 480. Alternatively, you could use the 1 over d squared. And so how many times bigger was 5 to 10? In fact, it's not bigger, it's half. So I did 0.5. Half distance squared and 1 over that. Okay. And that's why it's times by 4. Does that make sense? Tricky stuff. As hard as it gets, though, by the way. Right, so can I please ask you to move on to get your page out, the one where you've got the driving question at the top. Please use full sentences for your answers. Make sure you show all your working out. And yeah, let's have a go. Driving question is, how do the factors affecting the rate of photosynthesis interact? Okay, start off please with the describe section. That is, describe the patterns shown on graphs of light intensity or carbon dioxide concentration. Second section, explain, is explain the patterns shown on graphs of light intensity or carbon dioxide concentration. I would like you to do temperature as well. I might be able to add temperature in there as well. I'll go through the answers for temperature as well. So describe the patterns shown on graphs of light intensity, carbon dioxide, and temperature. You need to know those graphs. Use. You need to be able to explain the meaning of the inverse square law for photosynthesis and predict the rate of photosynthesis using the inverse square law. So those same questions, but I've just separated them out into boxes, which will allow me to put this table down. And using the fact that at 10 centimeters, there are 200 bubbles per minute, I would like you to figure out what the rest of them are. Give yourself between 10, 15 minutes, perhaps longer if you want the challenge on the last one. Work your way through them, start on orange, then green, then yellow, then blue. As always, make sure you're using the keywords in your answers. Good luck, year 9. Difficult stuff. We'll go through the answers in a minute. Good luck. Okay, welcome back. Please use a green pen to mark your answers. Give yourself ticks if they're right. If you're missing information, please use the green pen to fill in the missing parts. Right. For light intensity and carbon dioxide concentration, you have a graph that looks like this. If you haven't got it drawn, draw it now. Draw it. Steep gradient, levels off. For temperature, you see that slow increase gets to the optimum temperature and then a quick decrease after that. Draw this graph as well. Pause the video if you need extra time. Okay, actually what is going on? Light intensity, the rate of photosynthesis increases with increased light intensity at first, then increased light intensity no longer makes a difference. This is because at this point, either temperature or CO2 concentration are the limiting factors. Carbon dioxide concentration. The rate of photosynthesis increases with increased CO2 concentration at first. Then increased CO2 concentration no longer makes a difference. This is because at this point either temperature or light intensity are the limiting factors. And with temperature, a little bit different. To start with, increased temperature increases rate of reaction all the way up to the optimum. For most plants, this is around 40. After this, the plant will become damaged by the increased temperature and the enzymes denature and the tissue is damaged. And so you get decrease. Those are your explanations. It might help if you just put them right next to the pictures that you've got. It's up to you. Right, what is the inverse square law? Well, if you've just got this bit in black, if you haven't got this bit in bold, please can you just copy that in for now? The inverse square law applies to the relationship between light intensity and the distance from light source. It states that light intensity is proportional to the square of the distance of the light source from the plant. For instance, the light intensity at twice the distance away is spread over 4 times the area. The energy at 3 times the distance is spread over 9 times the area. You're not expected to draw the diagram, although it does help explain it quite nicely, I think. And then finally, let's have a look at those maths. Okay, so you need to predict, so this is the objective, being able to predict the rate of photosynthesis using the inverse square law. Well, the first one I decided to do is 20 centimeters. This is double what you started with. 10 centimeters to 20 is doubled, so 200 to mystery number is divided by 4. 200 divided by 4 is 50. Another way you could think of that is that the distance is doubled, so 1 over 2 squared, or 1 over 4, is a quarter. A quarter of 200 is 50. The next one up, I've done between 10 and 30 centimeters is tripled. So again, times 1 over 3 squared, or 1 over 9, divided by 9, is 200 divided by 9, 22.22. 15, a little bit difficult. There are a couple of ways to do this. One way, you could work back from 30 and say that, well, 30 to 15 is halved, so 22. So whatever 15 was, a quarter of that would be 30. So times 22.22 by 4 will give you 88.88. That could work. Alternatively, you could know that between 10 and 15 is times by 1.5 or 1.5. So what you do is 1.5 squared, which is 2.25, and you divide it by 2.25. 200 divided by 2.25 is about 88.8889. Okay, so similar numbers, both ways of doing that. The last one, I've decided to do it like this. So the distance is times by 5 from the first instance. So 10 up to 50 is times by 5. 5 squared is 25, so you do 200 divided by 25. 1 over 25, which is 8. So there we go. That is why I have these answers. Not easy at all, was it? Well done, anybody who got all the way to the end. Seriously, that must require quite a lot of concentration, especially with all the distractions at home, etc. Right, I'm very proud of us getting to the end of this one. Well done today, seriously. Please can you make sure you've got your today I've achieved sentence written at the end. If you've got to the use section, please can you write today I've achieved use because I can explain the meaning of the inverse square law for photosynthesis. Make sure you've got your becauses. Please pause the video, write that now. Okay, again, congratulations for getting to the end, guys. Seriously, not easy, that one. Again, as always, you've got 10 questions on your Show My Homework quiz. Make sure that you get those completed. That will just test you on various bits and pieces from these objectives. Okay, cheers very much, guys. I will see you next time. Bye for now.